Naughty Science coming to you live. Or maybe not so live. Maybe like pre-recorded cyberspace live. Whatever live you're coming from, please enjoy this video. Make sure you subscribe for more content. Thanks. The book I used was AP Bio Investigates from APSI. What you'll need is 2 clear cups. Got these from Walmart. Masking tape for labels. A syringe. Spinach! Cool. Distilled water. A light bulb. Hole punch. Dish soap. Baking soda. A measuring cup. A teaspoon. So we're going to take a teaspoon of baking soda into 300 milliliters of distilled water. We're going to stir it up till it's all dissolved. This is going to be our with CO2 water. So this is the most time-consuming part of this lab. You are going to hole punch 20 leaf discs that have to be perfect discs, no edges. You also want to stay away from that middle rib of the spinach because there's not much going on there in the sense of photosynthesis. It's just a lot of vascular tissue. You want the leafy bits. I highly recommend using an old school style hole punch. This thing was absolutely terrible for this. It took me way too long. But good old Walmart, right? So hole punch, hole punch, hole punch. Be very careful with these little discs. If you manipulate them too much, if you squish them too hard in your hand, you're going to damage them. You want them in as pristine a condition as you can get them. So make sure you empty the little container that holds the paper cuts, the little holes, regularly so that they don't get smashed up in there. Woohoo. So here's my 20 little discs for each variable. Okay, so now we're going to water down some dish soap, and the soap is used almost like a buffer to break up the lipids that are inside the cell membranes of the mesophyll, so we can get all the gases out of the spinach leaves before we begin our experiment. So what you do here is you just I put like a milliliter of the soap solution into our first, this is the control. And you have to be really careful not to get this sudsy, because if you put too many bubbles in it, the discs could get trapped and your experiment will be a flop. So that was our control. Notice I put that used pipette out of the way. I'm going to grab a clean one to do the next one to avoid any cross-contamination. You don't want to throw off your results. So once again, I just filled up my little pipette. This one I accidentally squeezed it a little fast, so it did suds up a little bit. So what I did to fix that was I kind of squirted everything out of the pipette and used it to gently stir, and then I sucked out the bubbles that I could get a hold of. So now we're going to take our discs and place them into our syringe. So I pulled the plunger off of the most complex syringe I've ever used in my life. Once again, Walmart. To be fair, it's for baby medicine, so it's like really precise and it has different measurements. I just wanted a basic syringe, but that's all they had because I did this at home, not at school. Alright, so you put your discs in there and you just try to get as much of that air out as you can without actually squishing the discs into the bottom. And like I said, this one was a pain because I had to screw the back on. And what I didn't realize is, you'll see it when I pull it, only lets me take in whatever measurement you twisted the little thing on at. It's the whole thing. Okay, so here I'm just going to take up some of this is the control. I do the control first because it's just distilled water and soap. So if I reuse this syringe to do the leaf discs again in the next one, I'm not going to throw off the experiment because it's just distilled water. It might affect a little bit of a concentration on a microscopic level, but for the sake of the lab, it will still work if you use the same syringe. Just do the control first. Alright, so here's the trick that everyone struggles with. So what you do is you actually put your finger over the syringe after you get all the air out, and you pull back gently, and then you push forward. And that push forward is what really gets all of those gases out of the leaves. And you can actually see them squirt out all over the place. And you want the discs to sink. Once they're sinking like this, you're done. Don't overdo it because you can damage those leaves. Alright, so I'm just going to soak up just a little more water from our experimental or control tube. So cover the end, and I put a little water in it so I can swish these guys out without having them get stuck to the sides of the syringe. So there we go. Cool. So there's the control is ready to go. So put the lid on it, and then we do the exact same thing to the next, to the other variable, your experimental. So this one I did show again because I figured out how to get the biggest amount, the 5 milliliter syringe to work. And I also did a little bit of a slow-mo on the squeezing of the mesofill to get it out. Okay, so I got all my discs in there, I got all the air out, all the way out. So I'm gonna pull and then push. Tap, 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 any bubbles out. See, we still got some that are floating around in there. We want them all to be sinking. So here's slow motion. I pull, and then I push. It's almost like you're knocking the wind literally out of these discs when you do that. And that's what got them all to sink in about 2 tries. So now we're ready to go. Okay, so we have our lids are on, our containers are labeled appropriately. So now we are ready to put them under a light source, and you want to make sure that your light source is centered, that there's not one getting more or less light than the other one. So for the next, I think 10 minutes, it's just this timer sped up. This experiment did take 20 minutes. I sped it up for the sake of my sanity and yours. Please feel free to fast forward. At the end of the experimental portion, I do a run through on the data and how to make a graph in Excel, which is probably the thing that frustrated my students the most when starting lab reports. So I hope this helped. Okay, so we watched a bunch of spinach discs float in water. Cool science, bro. Now you've taken all this random numbers and random data, and we need to put it into a digital format to turn it into a graph. So collect your data that you wrote on in your science notebook or a loose piece of paper or your forearm, whatever you did to collect your data, we're going to put it into a format that is acceptable to be turned in and put into a lab report. So I wrote mine on Post-it, so I would just went in and typed it all through here. I put 2 columns and labeled them appropriately with the minutes that I used. So what we're going to do is you're going to take the top value from your first set of data. So we're going to hit Shift and the right arrow and then click the down arrow all the way down to the end of the data. And then we're going to come in here to Charts and we're going to do a line graph. So that gives you just a very simple, straightforward graph. But notice it has no axis labels, it has no title. This would not be appropriate to incorporate into a lab report because doesn't mean anything, right? Because you're assuming the person reading your lab report doesn't know the experiment you did. So what I do is I come up here into these quick layouts. These saved me in college. So I always pick, I think it's this one right here. Yes, this one right here because it gives you the opportunity to label everything. And I hate these lines, so I just click them and you just hit delete and they go away. So if you right click and you say select data, it allows you to give your series. Your series are your categories of data, so you just give them names. So series 1, it's reading, so this is with CO2, whoop. Okay, and this is without CO2, okay. And I know that's not a subscript, it drives me nuts too, but time is money, right? So here, because we did this quick chart layout, it's just a matter of clicking on these and giving them appropriate titles. So this would be time in minutes. This would be your number of discs, number of floating discs. Because it's a type of title, a label, you always want to capitalize. These students get real tired of me being finicky about their graphs. And then just rate of photosynthesis, or cellular respiration, I guess. Photosynthesis for a big idea too. If it sounds like there's gunfire going off outside, it's July 3rd, so they're shooting off fireworks next door to me. I'll go out and watch them in a second. All right, so rate of photosynthesis, I don't know, blah, blah, blah. Okay, so give it a beautiful title, all that good jazz. And here you have it. And if you make this, slide it around, you can make this a little larger. I think it looks nicer. And if you double click this, you're able to kind of customize what you see. So I like ticks on my numbers. I feel like you're able to kind of follow the numbers better. But got the little ring of death right now. Come on. Come on, computer. Okay. So I think outside is how outside. And then minor tick marks. Now we don't need them. All right. And then you can customize this however you want. Just keep it simple is my advice for making these. I know you can go crazy in these programs and change the font and do all this crazy stuff. Don't do that because all it does is make it harder to read and it's not professional looking. So keep it clean. You can also change these lines because I know if you're printing it out, it's going to be in black and white. You can change the line to be broken up. So if you come up here to, I think it's in weights, style, you can kind of change it into like double lines and stuff, so it looks different even if it's printed black and white. So there you go. I hope this helped you. I hope this lab was beneficial to you, especially if you missed class. This is a nice way to, still get the data and still get the content that we covered while you were gone. So please reach out to me. Let me know if you need any help or you have questions. I am by no means a computer expert. This is just through trial and error what I have figured out and what works. So until next time, thank you very much.